Welcome back to the deep dive. We've spent years talking about AI that excels at finding answers.
Mm-hmm.
You give it data, you give it a goal, and it optimizes the heck out of it.
Right.
But today, we are jumping into a new frontier, an AI that doesn't just find answers, it actually helps define the questions.
And that is the absolute game changer here. For decades, the fundamental bottleneck in complex scientific discovery has been, well, twofold.
Okay.
First, the sheer size of the solution space. It's just combinatorially massive. And second, the fact that experimental validation is incredibly high cost.
In both time and money.
Exactly.
So, okay, let's unpack that central problem facing AI in the lab. We've seen AI agents that can optimize results brilliantly.
Oh, absolutely.
But they always rely on objectives handed down by a human. I tell the system, maximize molecular stability and minimize toxicity, and the KI runs that loop perfectly.
But that's the thing, isn't it? That reliance on fixed human-provided input is the fundamental flaw. Because those objectives are almost always imperfect proxies for the true scientific challenge.
Imperfect proxies.
Yeah, a human scientist might aim for high potency, but they might completely overlook a practical constraint, like, I don't know, how hard the final compound is to actually make.
So you get these agents just optimizing a static and often flawed definition of success.
Precisely.
The system does exactly what we tell it to do, even if what we told it leads to a practically useless result.
Right. And that brings us to the core of today's deep dive. The scientific autonomous goal-evolving agent, SAGA.
And our mission today is to really look at the source material and understand how SAGA overcomes this problem of fixed objectives by automating the objective design itself.
And the challenge it faces is enormous. Traditional optimization agents just struggle so much because the scientific landscape isn't a smooth hill.
It's what? A mountain range.
A vast, rugged, multidimensional mountain range covered in fog. And you're trying to find the highest, most perfectly accessible peak.
I think the best way to understand this failure is through a concept that translates terrifyingly well from the AI world to science: reward hacking.
It's fascinating how that manifests in this context. When those objectives are fixed and rigid, the AI, its evolutionary tendency is to exploit any gap or loophole between the model it's using and, hard reality.
So you mean the system finds solutions that maximize the predicted score,
Yes.
which makes the internal model look great, but it completely misses critical practical constraints that a human scientist would spot immediately,
Exactly. You end up generating these compounds that have incredible scores on paper.
but are basically useless in reality.
Totally. All because the AI hacked the proxy objective. It's the ultimate example of unintended consequences. If you only score for, say, avoiding toxicity, the AI might propose a gigantic polymer,
Which is technically non-toxic.
but way too large to ever be absorbed by the body. It achieved the goal, but the output is meaningless.
So what's often overlooked is that figuring out the correct objectives in the first place, that's its own search process.
That's the meta-search problem. And because it requires such high-level insight, that has historically been the true bottleneck. It's all been manual trial and error by experienced scientists.
The scientist looks at a failed experiment and says, wait, we maximized X, but we forgot about constraint Y. Let's add a penalty for that.
Right. SAGA seeks to automate that intellectual leap.
That makes sense. So the biggest hurdle isn't just iterating through solutions. It's figuring out how to set up the problem correctly from the start.
Yeah.
So how does SAGA actually structure itself to do this?
Well, SAGA's solution is this bi-level architecture that feels quite familiar. It's designed to mimic the concept of thinking fast and slow.
That's a powerful analogy. Okay, let's start with thinking fast, the lower level.
That's the optimizer. This is the workhorse. It uses established methods, like genetic algorithms, to just quickly and massively search for solutions that maximize the current set of objectives.
It's all about speed and quantity within a defined box.
Exactly. It's excellent at its job, but it's essentially mindless about whether the objectives themselves are any good.
So the real intelligence, the thinking slow, that has to live in the outer loop. And this is broken down into 4 distinct agents. First up is the planner.
The planner looks at the big picture goal, say, find a better permanent magnet, and it proposes specific new objectives based on the current progress and any weaknesses it's seeing. It sets the intellectual direction.
So if the planner decides on an abstract goal, like low supply chain risk, how does SAGA actually measure that? That seems incredibly hard to automate.
And that's where the next agent comes in, the implementer. This is where it gets really sophisticated. Its job is to take those abstract ideas and convert them into executable, quantifiable scoring functions.
Okay.
And here's the key. The implementer can actually write new runnable code, a new Python script, for example, to operationalize that objective.
Wow. So it's not just following instructions, it's building the tools it needs to define success.
That's it. So once those new scoring functions are implemented, they feed back into the optimizer, that fast inner loop.
But the whole system depends on the final agent, right?
Yeah.
The analyzer.
The analyzer is the ultimate critic. It examines the results, looks for failure modes, and synthesizes insights. It doesn't just celebrate a high score, it looks for systemic issues.
Like what?
Well, it can detect problems like a negative correlation between two scores. For example, as your potency goes up, your ability to synthesize the molecule plummets.
Oh, that's a bad trade-off.
A terrible one. The analyzer spots these trends, guides the planner for the next iteration, and ensures the system learns from its own failures.
And I like that SAGA doesn't force full automation. The source material highlights these 3 collaboration modes.
Yeah. The co-pilot mode is the most interactive. The scientist works closely with both the planner and the analyzer.
Then you have semi-pilot mode, where the human is a bit more hands-off.
Yeah.
They just give strategic feedback to the analyzer.
And finally, autopilot mode. That's fully autonomous. Ideal for these large-scale explorations once a problem is really well-defined.
I think to really grasp this, we have to look at the outcomes. Let's start with medicine. Designing new antibiotics.
Right. So the focus here is designing novel inhibitors for Klebsiella pneumonia.
A critical priority pathogen, I believe.
Very critical. And designing drugs for these gram-negative bacteria is notoriously difficult. Their cell walls are just incredibly tough to get through.
And the sources show that the old, fixed objective approaches failed spectacularly.
They really did. You look at a baseline agent like AlphaVolve, it was tasked only with achieving high K pneumonia activity.
And it did, right? The scores were great.
The scores were fantastic, but it suffered a catastrophic drop in medicinal chemistry quality.
This is that reward hacking we talked about.
Yeah.
It designed these unrealistically large, undrug-like molecules.
I mean, the system was so focused on that one score that it built molecular monstrosities. The analysis showed that one of the compounds was, and this is a quote, basically soap.
Basically soap. You can't put that in a patient.
You cannot. But it technically got a perfect score.
It's the perfect example of how blind a traditional optimizer can be. So how did SAGA, in autopilot mode, avoid this?
SAGA started with the primary goals, potency, low toxicity. But the analyzer agent immediately jumped in. It analyzed the entire population of molecules, and it spotted a clear trend.
The negative correlation.
Negative correlation between activity and drug likeness. It recognized the trade-off. It was basically telling the planner, we are trading practicality for predicted potency, and that's a bad deal.
So what did it do?
It did a granular structural analysis and noticed a huge compositional issue. Too many metabolically labile groups.
Which, for our listeners, means...
These are chemical features, like primary amines, that the body breaks down extremely quickly. The agents were designing drugs that would be destroyed before they even reached their target.
What an incredible piece of automated deduction. So SAGA saw the problem molecules that are soap and that break down too fast, and what was its autonomous fix?
It immediately added new scoring functions. Specifically, the quantitative estimate of drug likeness QED and a synthesizability score.
So it started penalizing structures that were too complex or didn't look like known medicines.
And by cycling through that loop, SAGA successfully generated novel antibiotic candidates that hit the drug likeness and activity sweet spot.
And crucially, these were novel structures, but they had similar commercial analogs, suggesting they were actually promising for real-world validation.
It grounded the theory in chemical reality.
So let's see how this generalist framework applies elsewhere. What about inorganic materials design?
This is a classic multi-objective problem. SAGA was tasked with designing new permanent magnets, but it wasn't just about magnetic strength.
There's another constraint.
A massive one, minimizing global supply chain risk.
So the objectives were high magnetic density, but also a low supply chain risk score, the HHI score.
Right. And for our listeners, an HHI score below 1,500 means the raw materials aren't cornered by just one or two countries. It minimizes geopolitical risk. You're designing for strength and sustainability.
How did it handle that?
It used copilot mode here. It optimized for magnetic density first, found strong structures, but they all used rare Earth elements, high HHI scores.
Not good.
So the human collaborator guided it to prioritize the HHI score next. And within just 200 high-fidelity calculations, SAGA identified 15 novel, stable structures. That significantly outperformed a specialized model, Mattergen, which only found 11.
It learned that a strong magnet you can't reliably source just isn't a good solution.
And you see that same principal discovery in designing superhard materials. The analyzer noticed that over 90% of its best proposed crystals contained essential light elements: boron, carbon, nitrogen, oxygen.
Which seems like common sense, but for an AI to learn that.
It's a huge step. It aligns perfectly with what humans know. You need those light elements to form the strong covalent bonds that make materials superhard. SAGA was rediscovering first principles.
Let's shift back to biology. Functional DNA sequence design. The goal was designing specific enhancers for a liver cell line.
And again, the classic failure mode. If you just optimize for a generic expression predictor, the sequences you get are, well, generic. They lack the biological specificity needed to work in a liver cell.
They generalize poorly.
Very poorly.
Okay.
So SAGA had to learn the biological rules of the liver. It iteratively refined its objectives to include things like transcription factor, motif enrichment, and sequence stability.
It was optimizing for biological plausibility, not just a raw score.
And the results showed a nearly 50% improvement over the baseline. But more importantly, the sequences it designed successfully recovered known liver-specific transcription factor motifs, like HNF4A and FOXA1.
Which confirms it had actually learned the liver's regulatory code.
It really did.
So across chemistry, physics, biology, the core takeaway seems to be that SAGA is an automated editor of the scientific hypothesis itself.
And this final application takes that practicality into applied engineering, designing chemical process flow sheets from scratch, a highly challenging task.
Like separating a butanol and water mixture.
For example, when baseline agents were trained only on product purity, they designed flow sheets that were cluttered with unnecessary unit operations.
So the product was pure.
Fantastically pure. But the process to get there was practically inefficient and way too expensive to build. The agent wasn't being penalized for complexity or cost.
Classic reward hacking in an industrial setting. You hit the score, but you blew the budget.
And SAGA's analyzer was designed to catch exactly this. It saw the system generating these technically successful but commercially useless processes. So it autonomously introduced new objectives.
What kind of objectives?
Things that an engineer cares about. Process complexity penalizing extra equipment, capital cost minimizing expense, and material flow intensity penalizing excessive recycling or tiny unstable streams.
So SAGA didn't just design a process. It designed a smart, practical, commercially viable process.
It balanced purity with cost and simplicity. And that leap enables automation that results in flow sheets an engineer would actually approve.
It's really the ultimate generalization of the framework, isn't it?
It is. Across all these domains, SAGA shows that objective formulation needs to be a dynamic, autonomous process. It directly addresses that core limitation of fixed proxy objectives.
It really solidifies that thinking fast and slow model. The inner loop, the optimizer, is the fast, expansive part.
Tirelessly exploring every solution.
And the outer loop, with the analyzer and planner, is the slow, critical, analytical part. It's the scientist questioning the premise.
Evolving the objectives based on failure, it mirrors the real scientific method far more closely than anything before it.
But we have to acknowledge the limitations.
Yeah.
SAGA's current success really depends on problems where objectives can be computationally verified.
That's the current edge, yes. For problems where the only validation is a messy, real-world experiment, SAGA needs extensions.
Like what?
The researchers suggest coupling it with fully autonomous lab-in-the-loop systems or relying on continuous human feedback to close that final gap between prediction and messy reality.
So, if AI can evolve its own goals, if it can look at a problem, try to solve it, and then decide it needs to change the very definition of success to get a valuable result, we have to ask ourselves a question. What complex, multifaceted problems, the ones we currently think are unsolvable because of competing constraints, will be the first to fall? The most critical question in accelerating discovery isn't just how to find a solution, but whether we've finally figured out what to look for.
A powerful thought to end on, indeed.
That's all the time we have for this deep dive. We'll see you next time.